Dr. Srujan Marepally on mRNA and nanotechnology.
A very good afternoon, everyone, and I thank Dr. Sudipta and the organizers for giving me this opportunity. Firstly, I'm a non-existential creature in pancreas because I don't work on pancreas. I work on lipid-mediated nucleic acid delivery. But when I told this to Sudipta, he told that you are a delivery guy, you can deliver anything. So I tried to entertain you in the next 12 to 15 minutes how nucleic acids can be delivered through mRNA and how this field can be also extrapolated to pancreatic research. Outline of my talk is, first I'll talk about emergence of mRNA as a therapeutic molecule, and then a brief introduction about lipid-mediated nucleic acid delivery, and then some of our work happening in our lab, like liver-specific nucleic acid delivery for hemophilia, and a few attempts towards a pancreas-specific delivery I brief about it. So gene therapy, the concept is very simple, like using genes as a medicines to treat diseases, but this is not as simple as it is, because when we look at these are cleaved by nucleases, and since DNA is also nucleic acids are negatively charged, the repulsion from the cell membrane. So there is a need of vector. So the world is still searching for an ideal vector. The first and the best so far, the in vivo gene therapy vector is the AAV vector. Our collaborator, one of the pioneers in AAV therapy, Dr. Arun Srivastava, calls this an awesome vector. This is one of the most successful vectors in gene therapy field. But there are challenges in this because this is a transient expression, exosomal, so we cannot use below 12 years of age until the liver is maturating. And then administration cannot be possible because it's like a vaccine. Once the vector is delivered, we cannot do that. So that's another challenge. And we have almost 90% to 95% in Indian population, and this is corroborating with multiple other studies, including China, Europe, and in US. So, but percentage varies, but no lesser than 80%. So 80% of the patient population is away from these, they are not eligible for the administration. So this is where the critical challenge lies. So these vectors are efficient in delivering the genes, but they are restricted to very less population. That's why I call this as a rare therapy for rare diseases. So this is well known that space tourism looks expensive, 0.2 to 0.3 million US dollars. This is a Blue Origin cost I'm talking about. Can you please guess what gene therapy product, any price tag? You feel this is cheaper. Going to space is much cheaper than having a shot of gene therapy product. Because we look at, this is Hemgenix costs $3.5 million. Zolgensma costs $2.1 million. Glybera, which is out of the market now, $1.6 million. This only got one shot after EMA approval, and Luxturna is $0.425 million US dollars in one eye. So these are critical challenges because their processes is extremely challenging. So that's where the vector design comes a very, very critical role. The viral vectors are very efficient in transfecting DNA, but they are not that efficient in the terms of scalability, and they have some immunogenicity. So somehow this can be overcome by non-viral vectors. But the critical challenge is their efficiency in delivering pDNA. That's why this field was there for more than 30 years to 40 years, but it didn't see light till 2000. So in early 2000 to 2010, the field is changed, and there when the biology exploded and RNA became a therapeutic molecule, this is hand in hand. Lipid nanoparticles have got the light. So if you look at the simple transfections, the mechanism of transfection, this is get endocytosed, and the cargo has to be released from the endosome, and then if it is a pDNA, that has to go into the nucleus and that is transcribed and then again comes back into the cytoplasm to translate the protein. So the viral vectors could do this job, like nuclear localization job effectively, but whereas non-viral vectors, lipid vectors, fail in that. So that's where the field was stuck. But when the RNA is developed as a therapeutic molecule, that became one of the potential strategies to overcome and get a therapeutic option for multiple diseases, disorders, whether infectious or genetic. So in a very simple way, gene therapy was there for a long time, but has a limited applications. But when the RNA developed as a therapeutic molecule, the journey shortened. It took a shortcut route that made a successful in multiple disorders. So I'll brief about the lipid-mediated nucleic acid delivery. This is a typical structure of a lipid nanoparticle, which has this is composed of individual lipids and then arranged in a spherical manner. This is a typical structure of the lipid. There is an ionized or ionizable lipid, it is a cationic lipid, which has a hydrophilic head group, hydrophobic tail, and a linker functionality. And the co-lipids consist of to mimic the plasma membrane, they have DOPE, DOPC, and the cholesterol as co-lipids. If you look at these are ionizable lipid, Delin MC3 is one already in the clinics. And I didn't put all, but there are certain like SM102 from Moderna, Actress Therapeutics, ALC3015 is from Moderna Pfizer's vaccine. So they're all used in the clinics. And the helper lipids are used is DOPC, DOPE, and the cholesterol. So siRNA is easy because something which is destruction easy, and this is 21 mud, and that is dimer. So it's very stable, and there are successful stories with siRNA therapy, like Onpattro, Vellore, and recently Leqvio. This is for hypercholesteremia from Novartis. These two are from Alnylam. So these inclisiran, which also got approval 6 months back in India for treating hypercholesteremia. And the other nucleus of drugs are like mRNA-based vaccines, which during COVID pandemic time, this got the limelight. Pfizer-BioNTech's vaccine or Moderna's vaccine. So they are also lipid-mediated mRNA delivery. And we've been working on lipid-mediated nucleic acid delivery from 2018, 2 years before pandemic. We're working on therapeutic angle where we try to deliver factor 9 mRNA to the liver. So for that we made a liver-detected nucleic acid, lipid nanoparticles. Why we chose hemophilia? Because hemophilia is a single protein defect disease where factor 9 or factor 8, if it is a factor 8 it is called A, it's factor 9 it is B, X-linked recessive disorder, and predominantly affects in males. There are therapies available, but they're short term, like clotting factor concentrates, which we need to infuse every weekly twice, and the monoclonal antibody emicizumab is effective but very expensive. And always when antibodies, again they develop antibodies against the antibodies. That's another challenge we have. And the long-term replacement is AAV-based gene therapy that we mentioned, like Hemgenix costs around $3.6 million. 80% of the world without access to hemophilia. The best part is like, even if you correct 5%, in our recent clinical trials, we found that even 2 to 4% correction also could make the patient transfusion free. So that's the best advantage with that. Even 1-2% you think is a success means that's a best model to take. So that's where we took since it's a low hanging fruit and we easily get some result, even if, so 35% is a success mark, but we need for success mark only 5%, means obviously we jump onto it. So that's the reason we chose hemophilia. So the short term versus the long term. So rarely used drug for rare diseases, AAV, versus very short, 3 to 4 days half-life. So we want a balance, like which is not that kind of AAV like once in a lifetime, or too short, like 2 to 3 days. So that's where another approach from Alnylam recently got approved 6 months back, fitusiran. So this is a GalNAc siRNA where it is modified with N-acetyl galactosamine so that it directs the cargo into hepatocytes and then cleave 81 to balancing the coagulation cascade. It has their positives and negatives. So there is a possibility of thrombosis as well. So that safety is studied, but still there are a few, that's why twice the clinical trials were halted. So we thought like, instead of that, if you can supplement the functional protein, like any other conventional gene therapy, that would be the best option. So this is an Arcturus Therapeutics data where they tried to inject in factor 9 mRNA in lipid nanoparticles, but it could only stay up to 7 days. Whereas Moderna did only, they also tried, but it went up to only 7 days. So there is a challenge to enhance the expression of mRNA. So that's where we took the challenge. And we had 2-level strategies, like we wanted to have a delivery system that has a sustained delivery, and we want an mRNA that is stable for longer time. So for that, to target into the hepatocytes, we chose asialoglycoprotein receptor mediated nucleic acid delivery. So we've been working on this for past over 1 decade, from 2007, and we identified our own modifications on that, like fitusiran did it. So we have patented our own, and this is what the lipid nanoparticle that we made. We call it a galtriazole, and then we made nanoparticles, and we just characterized those, like all are in 100 to 150 nanometers in range and has a positive surface potentials and have an uptake over 80%. And then you could appreciate that here the LNPs which are without galactose, that the biodistribution profiles are in multiple organs, whereas you can see galactose specifically into the liver. And this is the quantification data that we had. And the mRNA design that we took from is the albumin mRNA, because albumin is the largest protein produced by hepatocytes, and it has a good, we thought that since it has stability is high, so that mRNA we took from them, they truncated 5' and 3' UTRs, which play a very critical role, 5' cap and 5' 3' UTRs, and for poly A tail. These are very critical in the mRNA expression. And we also had base modification, and we had factor 9L mutant, which is a high functional variant that we chose. And we evaluated it. You could see there is a higher expression of albumin UTR of factor 9 in hepatocytes here. And we also have optimized the base modification, and we found that N1 methyl pseudouridine has the best translation in hepatic cell lines, and the fold chain is around 2 to 4 higher than the other uridine modifications. And then we injected into the animal factor 9 deficient animal. Then we evaluated the factor 9 expression levels. We could see this is almost 20% to 80% up to more than 40 days. So that's a fantastic preliminary result. With that, we are initiating our clinical trials very soon here. The summary is that gallinos enable efficient packaging of mRNA and deliver highly specific to the liver. We optimized 5' and 3' UTRs, which has a highly stable expression in hepatocytes, and N1 methyl pseudouridine is the best base modification in that. The expression of the functional protein is more than 30 days. So this serves as a platform technology not just for hemophilia but also for multiple monogenic liver disorders. And very recently, how we can correlate this, because the design of lipids are very critical in targeting in vivo organ specificity. So this is a very recent publication from University of Nevada, I think Las Vegas, So they, Bhattacharya's group, they demonstrated that this is incorporating cholecalciferol into the lipid nanoparticle system that directs cargo from liver to pancreas, and that with a higher specificity. So this is where one encouraging result. Couple of months back it got published. So in case if this is, but anyhow this is an early stage. If future research is needed, like how best we can, how specificity and without compromising the safety profiles, we can deliver the cargo into the pancreas. But there is a good beginning has happened, and we hope that we'll also look into pancreas-specific delivery. This is, I'm from here, CSCR, which is half a kilometer from here, other side of the road, and I thank my colleagues and students and collaborators and funding bodies. Thank you.
Thank you, Dr. Srujan. Any questions from the audience?
Excellent talk. I really liked it. Thank you for sharing your work. The question about the factor 9 data you showed. So what's the normal half-life of factor 9? Like albumin has a half-life of 21 days, 25 days.
Factor 9 is highly unstable. That's why it has to be infused every 3 days.
Every 3 days.
Yeah.
So have you attempted redosing after 37 days or 40 days when it came down? Did you see as efficient in synthesis as the first time?
That we are doing, but the one challenge with one which we did is that this is a C57 background mouse.
Yeah.
So even though factor 9 deficient, the challenge is that the redosing we are having challenging because of we are expressing human factor 9 in mouse model.
Yeah.
So we are working on immune compromised knockout model for that.
In terms of organ specificity, like you showed that when you added the GalNAc, that the liver specificity went up, and they added vitamin D3 and it went to the pancreas. So is this a hit and trial approach, or is there a logic behind the choices?
Usually in lipid nanoparticle design, it's a high throughput design, people vary these compositions, and based on the composition, even if you look at our strategy also, we varied multiple, like we made around 20-30 formulations and screened. This is how best you match the membrane of that particular cell, and also how they interact with because when you inject, the serum protein interactions are also critical. So what we even found that even a small variation in the formulation sometimes have a bigger effect. So yes, this is a hit and trial method. We need to screen large, but there is a kind of a set of rules like this. Say suppose if GalNAc that binds to ASGPRs, and if it is mannose that goes to dendritic cells. So those kind of set of rules are there in this field.
Very, very encouraging. Thank you for sharing.
Thank you.
I'll be curious because in the pancreas itself we have 4 cell types, and it'll be very interesting to see how ductal versus exocrine versus islet and all can be explored further. Thank you for sharing.
Thank you.
Thank you, Dr. Srujan. So